Hello everyone, my name is Miss Tanvi. Today we are going to start with a new part of immune system chapter, which is adaptive immunity. Adaptive immunity is rightly called as the body's elite defense system. Why is it called as elite defense system? Let's quickly jump into it. We have two arms of the defense of adaptive immunity. First one is known as the humoral immunity, which is mediated by B cells, and cell-mediated immunity, which is taken care by T cells. Now, if you can see the diagram below, there are differences between the B cells and T cells. Why do we call them as B cells and T cells? Let's take a look. The B cells, they are mainly originated and maturation site is bone marrow, whereas for T cell, they origin in the bone marrow and subsequently they are transferred via the lymph to thymus. And their main maturation happens in the thymus. Deployment, they circulate via the lymphatic system. For T cells, the deployment is via lymphatic system and peripheral tissues. The main thing that we need to know about is the receptor type. B cells, they have membrane-bound antibodies, whereas for the T cells, the receptor, they are of CD4 and CD8 types. Let's quickly move on to the next part, which is the antigen. The antigen is like the ID card. Any pathogen which enters into your body is going to have small molecules on its surface. These molecules are the ones that will trigger an immune response. Now let's take a look over here that you see this is a type of antigen. Now on this antigen you will find certain receptors. These receptors are nothing but antigenic receptors. These receptors tell the body that this is a foreign material which has entered our body and we need to eliminate it. Let's come to the next type, which is humoral B cell response. How this B cell works, what happens exactly. Before we move on, we need to know the meaning of this word humoral. The word humoral comes from the word humor, meaning some fluid in our body. Now we have two types of fluids in our body, mainly the blood and the lymph. Okay, so how they work, let's take a look. The B cell differentiate upon detecting specific antigens. So let's assume that this is our B cell. Okay, now this B cell, on its surface, it has certain Y-shaped proteins called as the antibodies. Okay, these are Y-shaped proteins that you see. They are known as the antibodies. Okay, I will quickly label them as antibodies. Now, when these antibodies, B cell containing antibodies, they come in contact with a pathogen. Okay, let's change the color of the pathogen. Okay, now let's say this is our pathogen. It came in contact with the pathogen. What is going to happen is, it's going to undergo some differentiation. It will form two types of cells. The first type that we have is known as the plasma B cell. And the second type is known as the memory B cell. Okay, plasma B cell and the memory B cell. This plasma B cell is going to secrete enormous amount of antibodies. Okay, it is going to secrete enormous amount of antibodies, which is through the humor part of our body, blood and the lymph. What happens to this memory B cell? This memory B cell is going to be like a guard forever in our body. Forever is an overstatement. It's mainly for decades. Okay, this is going to be guard cell in our body for decades. Guard cell meaning what? This memory cell will keep the recipe to eliminate this type of pathogen forever, and for decades it is going to go on. So that was about how any B cell is going to work. Let's quickly come to how antibodies look, what is the function, what is the structure. Now as you see, any antibody is going to be like a Y-shaped structure. Okay, this is how it looks. It's a Y-shaped structure. It has sites for antigen binding and these sites are very specific. So as I said, hundreds and thousands of pathogens enter into your body with different antigens. Our antibodies are capable of identifying them every time. Before we move on to cell-mediated type of immunity, we need to understand one concept which is about MHC, which is known as the major histocompatibility complex. Now, what are these MHC molecules? These are certain molecules that you find on cells. Okay, on all type of cells we have these MHCs, even your foreign cells and your own cells. They are of two main types. MHC1, which is found on all nucleated cell, which is present in our entire body. So this tells our body that, okay, this is my own cell. I don't have to attack this cell. This is my own self identity. Then you have this MHC2, which you find on APCs, which means APCs are nothing but antigen presenting cells. Okay, antigen presenting cells like macrophages. This ensures that the body knows that, okay, there is some foreign material that has entered, a foreign pathogen has entered. Okay, why is this part significant? You will understand it in the next slide. Why we need to know this part. Okay, let's come to cell-mediated, which is the inside job. Focus on intracellular pathogens, viruses, some bacteria. This involves direct cell to cell. And as we discussed earlier, cell-mediated immunity is mainly done by T cells in our body. T lymphocytes by our body. Okay. Let's quickly come to the type of T cells. In our body, we are having three main type of T cells. The first type that we have is known as the helper T cells. Okay, we also write them as TH cells. The cytotoxic T cells, we write them as TC cells. And the T regulatory cell, we also write them as small TR cells. Okay. Now, what is the difference between all of them? Now, as you can see, the major difference is on these receptors, CD4, CD8, okay, CD25. These are certain receptors which dictate their function. Okay, let's understand this. T helper cells, they have a T cell receptor, which is going to be common in all of them. Okay, T cell receptor, common. It has CD4 receptor. Its main job is system activation. It guides cytotoxic T cells and B cells to work. Helper T cell is more like a general in the body. Like in army, you have various types, like the main head. Okay, it's like a general in the body, these cells. They will tell other cells on how to work. Okay. The next type that we have is cytotoxic T cells. From the name itself, toxic, meaning it is going to ensure that eliminates any threat from the body. Okay, see, it's important for threat elimination. It is responsible for removing pathogens and destroying infected host cells. Cytotoxic cells are important because in our body every day there are hundreds and thousands and millions of cells which are dead. So to remove these dead cells, we need a machinery that is done by cytotoxic T cells. Anti-regulatory cell, regulation, meaning it's going to make sure that friendly fire does not happen, meaning self and non-self, that distinguishing characteristic is taken care by the T regulatory cell. It distinguishes self from non-self to actively reduce the risk of autoimmune disease. So as you know, there are certain autoimmune diseases. One such example is rheumatoid arthritis. It is mentioned in your books. During this disease, the body fails to distinguish between our own cells and the foreign cells, and it starts attacking our own cells. Okay, so it's very important that our body cells, we have our proper TR cells. The TR cells, on the other hand, they have CD25 as their receptor. The cytotoxic T cells, they have CD8 as their receptors. So far, these are the types of T cells. Let's quickly move on to the next part. The T helper cells. Now, how do these T helper cells work? As I said, these are the generals of the body, generals of the immune system. They will guide the B cells and the cytotoxic T cells to work. So let's take an example of how they exactly work. Okay, so if this is our one of the T cells, okay, this is our T cell. The T cell is going to have MHC complex over here, MHC complex. Okay, this MHC complex is going to bind maybe, let's say, to some antigen. Then this cell, which is our naive T cell meaning it has not taken up any function yet in our body. Okay, this naive T cell is going to get distinguished into three parts. Okay, either of three things will happen. Because of this cell, the helper CD4 cells, okay, if it has the helper CD4 cells, it is going to go and tell the B cell to secrete out antibodies, okay. It will stimulate the B cell and it is going to make antibodies. What another thing happens with the helper T cell, okay, second fate of this helper T cell is going to be where it is going to tell a macrophage. If you remember macrophage, these are nothing but monocytes in our tissues. It's going to tell a macrophage, and then a macrophage is going to release out cytokines, okay, they will release out cytokines. The third thing that can happen with any T helper cell is they will go and they will stimulate TC cell, okay, see over here, cytotoxic T cells, okay. This is how any T helper cell is going to work in three ways. Either first way, where it will tell a B cell, it will stimulate a B cell to secrete out antibodies, it will stimulate macrophage to release out cytokines, or it will either end up telling CT cell, TC cell to do its job, okay. Let's quickly move on to the next part, which we have is cytotoxic T cells. Okay, so now let's understand how cytotoxic T cells work. Cytotoxic T cells, as we mentioned previously, they have CD8 receptors on their cells. They are known as the assassins, meaning their job is to clear out any dead debris or any infected cell in our body, and they do this by apoptosis. Apoptosis is programmed cell death, okay. What is apoptosis? Programmed cell death, meaning every cell has a time limit to it. When that time comes, the cell is going to be reaching its senescence, meaning it's now old, and now the cell has to die. That is taken care by cytotoxic T cells, okay. So let's see how they work. They work mainly by releasing certain perforins, okay. Let's see the illustration now. Now if you see over here, this is our cytotoxic T cells. Cytotoxic T cell will have its own receptor, okay, which is the T cell receptor, and it will also have one special receptor which we mentioned, CD8 receptor. Now in this case, let's say we have one infected cell, okay, this is our infected cell, okay, infected cell. This infected cell, it will also have class 1 MHC molecule, okay, MHC1. Now what happens is, inside this cytotoxic T cell, they have certain proteins which are known as perforins. Now let's break down the name. Perforin meaning something where it makes perforations or it makes holes, okay. So this cytotoxic cell, when it comes in contact with an infected cell, it will release these perforins, okay. What will happen? These perforins are going to be released outside, okay. So see, these perforins will be released outside, and when these perforins are released outside, the infected cell will have certain pores in it, okay. It will have those leaky channels that are getting formed, okay. So these are the leaks, okay, because of perforins. I'll just quickly change the color, yeah. Perforins have acted on them, and now there are leaks in the entire cell, and because of this leaks, cell death happens, meaning our body has avoided a toxic cell, our body has avoided a dead cell, and that's how that clearing takes place, okay. So this is the function of cytotoxic T cells. Next part, which is differentiation on how these cells work, okay. A quick brief recap. So as we have seen, the B cells and T cells, they have different mechanisms. The B cells generally have a lock and key mechanism, whereas the T cells, they have a presenter type of mechanism where the MHC molecule plays a very important role. B cells, they have direct binding, and T cells, they have indirect binding. The T cells are blind to free pathogens. They only recognize antigens presented on the MHC molecules, whereas B cells, they capture free antigens which are roaming in your body, and that's how you are given protection. This, what you see over here, is a basic difference between the two types of immunity, humoral immunity and cell-mediated. Humoral is mainly body fluids, blood, serum, lymph. Primary mediators, we have our antibodies, and mechanism is the free-floating antibodies bind to antigens and neutralize them. And the cell-mediated type, infected host cells and intracellular spaces. We have our main players, T helper cells and T C cells, cytotoxic cells. They physically eliminate any pathogen or any dead cell or any toxic cell. So that was it, everyone. Thank you for joining.